looking at dinosaur fossils. The bones have been fossilized from dinosaurs. And these honestly look more like rocks to me, but they're, what do you have here? What are you studying?
Well, this actually is not a bone. These are fragments of a Triceratops horn.
Okay.
In 2012, the Creation Research Society sponsored Mark Armitage and I to go to the Hell Creek Formation in Montana, which is a very popular place for finding dinosaur bones, and we instead dug out a Triceratops brow horn. Now, it's just in crumbled pieces now, so we can't really, put it together and show you a horn. And what we've done, of course, is have to work with it where you actually destroy portions of it. But you have to recognize that inside that rock-looking structure are tissue, cells, and protein still there.
All right, so let's go back for a second, because if this horn or part of a dinosaur had been buried for millions and millions of years, you would not expect to still be able to see tissue. But are you saying that's what we're finding?
That is absolutely what we're finding. In fact, in a Nature Communication paper in 2015, they referred to it as common.
So is this unusual for those who have followed the traditional paradigm associated with when dinosaurs lived and when they died?
It would certainly be not at the least expected. In fact, Mary Schweitzer, who was the first one to really make this popularized, the first one to really get discoveries that were noticed by a wide range of scientists, she comments in interviews that she had her technicians repeat the study over and over and over again, simply because it's so difficult to understand how you could have this material still in a dinosaur fossil that is supposed to be 65, 70, 75, 80 million years of age. Because any competent biochemist knows that tissue, cells, proteins break down. They're not concrete, they don't just exist for eons of time, they break down. And in fact, they tend to break down fairly quickly depending upon the conditions. And certainly in Hell Creek, the conditions would be warm up, cool down, warm up, cool down. We found this horn, for example, just a mere 1 foot below the surface. Now, it was in very solid sandstone, so we had to chisel it out, but it was just below the surface. So there was no thermal protection by being deep in the ground. So it would have been very much subjected to fluctuations of hot and cold and hot and cold. And any biochemist can tell you that is the fastest way to destroy material. It's difficult enough to envision it surviving for 4,000 or 5,000 years, but 60 million years, 70 million years, see, that really becomes very difficult to make any kind of biochemical basis for how it could have survived.
Okay, well, let's talk now about what you do with these, okay? You've got these here. What would be your standard process of wanting to then examine?
Well, as you can see, these look like a rock. So what you have to do is you have to literally dissolve the rock away. So what we do is we soak the fossil material in a solution called EDTA. It's a very mild acid that will grab the calcium ions out of the fossil. And so you just literally dissolve the fossil. And what you'll have after you dissolve the fossil is the tissue will be remaining, because the EDTA won't dissolve the tissue.
So where are you now in this process? What would be your next step?
Well, some of these have just started, but the next step would be then we would take and we'd pour off the solution.
Okay.
Right now, I'm not worried about what's in here. I will collect that and analyze it at some point later. But right now, I'm just going to look at this.
What's left there?
Right.
Okay.
So then I'll bring this over to what we call a dissection microscope. As you can see, what we see is this is in essence dissolved Triceratops horn magnified.
Well, Kevin, what did you find then when you were looking at the sample and you actually found some tissue?
Okay, here's what we found. Here we have a piece of the horn that has been decalcified, like what I just showed you over on the bench. Mark Armitage was our microscopist working on this, and Mark then took a piece of the decalcified horn and put it under a microscope. See the fibrous material there? That's part of the composition of the bone matrix itself. But what's really of interest is see the white material here on the surface swaying back and forth? That's actual dinosaur tissue on the surface. See, this is not a solid fossil. This has got tissue characteristic to it. See, notice how it flexes back. And that's, of course, very interesting how you pull on it, flexes back, pull on it, flexes back. That's characteristic of tissue. That's what tissue would do. Now, Mark then was able to extract some very thin layers of elastic material away from the inner core of the horn. But he didn't have to decalcify the horn in order to do this. You can see it's stretchy, it's flexible. In fact, look, notice, see how it's stretching? It's stretching almost to twice its original size of what it was. See, this again is original dinosaur tissue that he's peeled directly from the fossil. There was no decalcification that he did first. See, this is how accessible this tissue was. He didn't have to remove any fossilized bone to get to this tissue. Okay, now, here is a light microscope picture of the tissue itself. You can see the texture of it. And in fact, see, notice the arrows, they're pointing to cells. These cells specifically are what we call osteocytes. Those are bone cells. They're involved in making bone, because even though we think of bone as a rock, bone is tissue. Bone is not a rock. In fact, in our bodies, bone is replaced about every 10 years. To keep it all fresh and the new matrix laid down and such. So it's constantly being changed, and that's what those cells do. And if you look at them then at a closer magnification, what we see then, this is using scanning electron microscopy, you see the extreme detail of the cells. See how well that's preserved. I mean, that doesn't speak for something that has been degrading or something that has just been in a non-pristine condition for 65, 70 million years. We would not begin to expect to see such enormous and elaborate detail. I mean, these structures are incredibly small. You know, this is our 20 micron bar here. See how small these structures are, still intact. And yet to see that kind of detail, then obviously the preservation process is surprising really to everybody. Yeah. But I think as creationists, we have a lot less to explain than someone trying to suggest that this is, 65, 70 million years of age.
Well, let me ask you this first, because, I mean, this is an incredible picture. And I just want to make sure we're not looking at fossilized material here, fossilized tissue. That's been replaced by calcium.
These are the cells.
These are real cells.
Correct.
Well, Kevin, I understand you published all of this work.
Yes,
is that right?
This work has been published. We've actually made the cover of American Laboratory. We also published in a journal called ACTA Histochemica, and that's a more technical article. It goes through and explains then what we did, how we processed the horn, and then of course draws some conclusions from that. Particularly the conclusions are that even though it's a horn, which is different from a bone, it still had tissue. Even though it was wet when we pulled it out of the ground, it had what we call matrix, which is another word for mud. It still had tissue, so just a different specimen than what had been analyzed before. No one had done a horn before. Now we start getting closer, and what you're going to see then is you're going to see little pieces of tissue there, and that's what you're going to pick off and then put under the higher powered microscope. Okay, and then under the higher power microscope, see, now we look at the tissue itself, and there's the cells.
Oh, it's fascinating.
See, and then there's another. And like I say, those are very specific osteocytes. Now, those osteocytes have a very unique structure. Okay, now we're coming into some of the texture of the horn itself. And see, this would be a blood vessel. And if you look, you see, this is dissolved away. This would be the interior of the horn. And you can see all the detail that'd be left in there after you take the calcium away. And then there, of course, is an osteocyte.
So that has to have shaken up the scientific community. What's been the response of all of this?
The initial response, when Dr. Schweitzer first published her work, which is what became very popularized in 2005, it generated a lot of response that previous papers had not generated. And there's kind of a question of exactly why it generated so much response. But in 2005, her paper was published in the journal Science, which is a very broad distributed journal, very highly respected journal. Color pictures, you can't minimize the impact of color pictures. So you had people that would be more biochemistry and biology backgrounds that maybe for the first time paid attention to this. So that generated, I think, some not only publicity for it, but certainly some controversy.